Hello everyone, my name is Eric Ron, and today I'm going to be presenting a paper entitled BMP Protein Mediated Crosstalk Between Inflammatory Cells and Human Pluripotent Stem Cell Derived Cardiomyocytes. This paper was published by the Fretz Group in 2017 and was published in the Journal of Engineering of Regenerative Medicine. And here's just an overview of what I'm going to be going over in my presentation. Initially starting with a background on myocardial infarction. So a myocardial infarction is going to be irreversible tissue damage due to prolonged ischemia, which is going to lead to a deprivation of oxygen and nutrients and lead to an accumulation of waste. In this image, you can see two infarcts. One is on the upper right-hand side where there's necrotic tissue. This was actually a lethal cardiac rupture. However, there was a previous infarct that you can see on the middle part of the heart, and this is indicative of an old infarct. So histologically, this does have significant changes in cardiomyocytes. On the left-hand side, you could see a healthier cardiomyocyte where you have healthy mitochondria, small lipid droplets, and then T-tubules, and all of the bands are well defined. However, in the post-MI heart, you get accumulation of lipid droplets and mitochondrial calcium phosphate deposits. The three main causes of MI are vessel occlusion, LV hypertrophy, and coronary hyperspasm, with vessel occlusion and LV hypertrophy being the dominant factors in MI. Next, I would like to just discuss quickly some tissue engineering strategies that have been done to help post-MI hearts. So this paper focused on human mesenchymal stem cells and their ability to repopulate cardiomyocytes. This is already being used in clinical and preclinical trials, and they are showing promising results. The reason that they are choosing these mesenchymal stem cells is because they do have multi-lineage potential, immunomodulatory properties, and they secrete important factors, specifically ones that support angiogenesis and ones that modulate the inflammatory environment. So the ideal bioengineered cardiac patch will be able to replace lost cellular mass, rescue heart function, and decrease the remodeling seen post-MI. However, there is still much to learn about the mutual interactions between cardiac repair cells and their inflammatory environment. There also are limitations specifically in retention of stem cells, the stem cell survival, the engraftment of the stem cells, and really what's detrimental is the inability to restore loss of cardiac muscle after injury. So next I want to discuss the role of the inflammatory response in cardiac repair, specifically looking at two different types of macrophages, one being the pro-inflammatory M1 macrophage, which is seen early on in an infection, and then later on differentiation into M2 macrophages, and these are dependent on specific chemical messages that are both concentration dependent and time dependent. In the lower panel, you can see two different slides. The first one is 24 hours after reperfusion, and then panel B is 7 days after reperfusion. The cells that are stained in red in the first panel are the neutrophils and monocytes, and then there's still some antibodies that are present for the macrophages. However, in panel B, 7 days after the MI, we pretty much only see M2 macrophages. This reflects repression of the acute inflammatory response. So one of the big advantages that I saw with this paper is that they are looking at the bone morphogenic proteins as chemoattractants rather than classical chemokines that have been shown to influence macrophage migration. So bone morphogenic proteins are normally involved in the development of the cardiovascular system. They're part of the transforming growth factor B superfamily and comprise more than 20 members. In the developing cardiomyocyte, BMP4 is released from the ectoderm and promotes the transition of pluripotent cells to the mesoderm stage in a dose and time-dependent manner, while BMP2 is secreted from the endoderm. In the first figure, I've shown the signaling pathway for the BMP proteins. BMP4 and BMP2 are known to both have affinity for the type 2 receptor and then activate type 1 receptor subsequently. And then in the adjoining figure, I've shown why it is advantageous to study BMP molecules in the development of cardiomyocytes, because you can see that they are influential in a lot of the steps going from a stem cell into a functioning cardiomyocyte. And really, this brings me to the entire objective of this paper, which was to really quantitate and understand what molecules are being secreted by the macrophage and what influence those have on the cardiac repair patch, and also what molecules are being secreted by the repair patch and how those influence the macrophage. Next, I would like to just discuss some of the methods that were used in this paper that help them figure out what the consequential fates of BMP and the different macrophage subtypes had on both cardiomyocyte yield and proliferation. So first, I'm going to show you how they differentiated the stem cells into cardiomyocytes. This was done over a 7-day process and was confirmed via expression of GFP protein, as you can see in the bottom left figure. This specific cell reporter line only expresses GFP when the cells are differentiated into cardiomyocytes, and from that they were able to make the histogram shown in the bottom right of this panel, where they're showing the count of troponin positive cells. I also want to highlight that this stage 5, where I have the star, is where they collected and isolated the CM maintenance media that they were using throughout the rest of the study. Similarly, they needed to isolate human mononuclear cells and then polarize them into the macrophage subgroups. This was done as seen in the profile shown to the right. And again, just highlighting that at day 7, this is where they collected and isolated the CM conditioned media that they will use for the rest of their experiments. So they confirmed the polarization of these monocytes into the specific macrophage subtypes by looking at specific markers. Specifically, CCR7 and HLA-DR are M1 pro-inflammatory markers. CD206 is an M2A marker, and CD163 is a pro-healing marker. The histograms on the right confirm that they indeed were successful in polarizing their macrophages, and this was also shown via fluorescence microscopy in the lower images. In the paper, they did discuss that one of the limitations was keeping these macrophage subtypes in their differentiated state. And this was done through the addition of maintenance media. However, one thing that I found particularly interesting from their microscopy images is that there was a heterogeneity in expression of these specific markers as well. Next, a 3D inversion assay was used to quantify macrophage migration through conditioned media. In this, they tested all the subpopulations of the macrophages. They also used a BMP2/4 neutralizing antibody, as well as a BMP2 and a BMP4 standalone neutralizing antibody. They also had positive controls of recombinant BMP2 and BMP4. And for all these inversion assays, MCP1 and SDF1 alpha were used as positive controls because they are known to influence macrophage migration. So some of the results. So the first 3D inversion assay looked at the effect of human mesenchymal stem cells on macrophage recruitment. So in this diagram, its migration to the gel is shown as by the arrow. These images were obtained via confocal microscopy, and they clearly show that the M1 subtype has the most migratory potential of the macrophage subtypes. However, with addition of the cardiomyocyte maintenance media and conditioned media, you see an increase in both M2A and M2C recruitment. And also that the maintenance media and the conditioned media both influence macrophage migration greater than MCP1 and SDF1 alpha. So in the center panel, these are the depth maps from the migration shown in the previous slide. And then in the far right, there was the quantification of the subtype. The inferences that they were able to make is that CM conditioned media triggered an M1 macrophage migration more than CM maintenance media. They also did not find difference in migration of the M2 subtypes. And they also noted that the M1 macrophages migrate at a higher number and travel a further distance than the other subtypes. The star on the figure indicates that the M1 macrophages exposed to the CM conditioned media were the only ones to travel greater than 700 micrometers. Next, the authors looked at the contributions of cardiomyocyte derived BMP proteins in macrophage recruitment. So these were quantified at day 30, and they both quantified the release of BMP2 and BMP4 into conditioned media. The real-time PCR graph on the left gives you an expression level relative to glycerol 3-phosphate dehydrogenase, and then the ELISA will give you a secreted amount of the BMP molecules. In both cases, they found that BMP2 was expressed and released higher. So next they used the cardiomyocyte conditioned media to study the role of BMP2 and BMP4 in the recruitment of specific macrophage subtypes. And this is the same 3D inversion assay as they did before. In the first column, they have both the BMP2 and BMP4 neutralizing antibody, where you see little to no migration for all the different subtypes. With addition of the recombinant BMP2, you don't get a recovery in any of the migratory tracts. However, with the addition of recombinant BMP4, you do get recruitment of the M1 subtype. And these results demonstrate that cardiomyocyte conditioned media promotes the migration of M1 through a mechanism that involves, at least in part, cardiomyocyte derived BMP4. They also did a quantitative assay of the expression of the BMP receptors in macrophages, and for both the BMP1 and BMP2 receptor, they found a higher expression in the M1 macrophage. However, at the end of the study, they did describe that this could have transitioned during the differentiation process, and this is something that they would like to quantify later in further studies. Next, the researchers set out to look at the effects of macrophage-conditioned media on human mesenchymal cardiomyocyte yield and proliferation. So between day 25 and 35 of differentiation, GFP-H9-derived cardiomyocytes were detached as single cells and exposed to different macrophage-conditioned media for 4 days. After these 4 days, the cells were stained with a Hirsch stain that shows up blue and dyes the nuclei. And then they did fluorescent microscopy to quantitate this in the same way that they did before, using the histogram as shown in the upper right-hand part of the slide. The authors did say that there was no significant decrease in total cell number when exposed to different subtypes of macrophage CMs. However, in looking at the histogram plot, it does seem to me that the M1 macrophage did have a significant change in the number of cells and was able to proliferate more than the other cell subtypes. Next, they used troponin T to quantify the yield of cardiomyocytes. Again, they did the same fluorescent histogram calculation as shown in the upper right-hand corner. And the results that they showed an increased yield of troponin T positive cells when exposed to the M1 conditioned media. And this can be seen in both the histogram and in the immunofluorescence shown below. These results do indicate that M1 induced differentiation does increase the yield of troponin T cardiomyocytes. Next, they looked at the proliferative profile of the specific subtypes. This time, staining for troponin T in green and Ki67, which is a prolific factor, in red. In the plot shown in the middle, they are showing the troponin to Ki67 ratio fold change, showing that the M2A and the M2C have a higher proliferative profile than the M1 subtype. This was further confirmed by looking at another factor, the PDGFR, which is a chemoattractant for stabilizing periocytes. The way that I took this to mean in the paper is that this would be angiogenic factor. And as shown by the relative fold change in fluorescence, exposure to M1 conditioned media led to an increase in these PDGFR cells. Next, they set out to study the antithesis of what was previously done, where now they're looking at macrophage-derived BMP-mediated signaling in embryonic stem cell cardiomyocytes. So BMP2 and BMP4 are expressed not only by cardiomyocytes, but also by all of the macrophage subtypes studied. This was confirmed via immunofluorescence analysis in the top right corner, and was also quantified via ELISA. And in both cases, M1 macrophages showed the highest expression of both BMP proteins, and this can be seen in the fold difference of the subtypes comparatively to the M0 in the left-hand figure. So to analyze whether BMP is active and sufficient to act, this was analyzed via Western blotting of macrophages, and also a densitometric quantification of the SMAD1/5 normalized to SMAD1, which are the downstream signaling molecules for the BMP pathway. So the results they found were that M1 conditioned media induced phosphorylation more than basic media or control. This effect was counteracted by the addition of neutralizing antibodies and recovered with the addition of recombinant BMP2 and BMP4. They also concluded that kinase P38 was not affected by phosphorylation, indicating the canonical pathway. Lastly, they looked at the contributions of macrophage-derived BMP proteins on cardiac repair cells. So the first image shows a decrease in troponin T positive cells in the presence of the antibody when exposed to the M1 conditioned media. However, you did see an increase in troponin C for the M2A profile and the M0 profile. The second lower image shows an increase in proliferation when the cardiomyocytes were exposed to the M1 conditioned media, supplemented with the neutralizing. Again, this was confirmed and quantitated using fluorescence microscopy. The results showing that recombinant BMP2 led to a significant increase of cardiomyocytes, while recombinant BMP4 resulted in a decrease in cardiomyocytes. While recombinant BMP2 did not affect the proliferative cardiomyocyte profile, however, cardiomyocytes that were exposed to recombinant BMP4 were less proliferative. The discussion section of this paper was more of a summary of the results. However, I will try and do my best to infer what I can from some of the other research that I was looking at. First, I want to just go over the main questions of this study, specifically being how do macrophages interact with cardiomyocyte repair patches, and how can these interactions be harnessed to design better patches with suitable characteristics. So following myocardial infarction, the injured heart does undergo an infiltration of neutrophils and monocytes. These will interact with the cardiac patch, first with a pro-inflammatory response and then later with a pro-healing response. These have been known to be done by certain chemokines, as shown below. However, this study was mostly looking at the role of BMP protein. I was a little disappointed that they did not compare some of the chemoattractant properties of BMP to any of these other known attractants. However, I think this is something that they could do in a follow-up study. Their 3D inversion assay analyzed the ability of the different macrophage subtypes to migrate towards the conditioned medium. And using neutralizing antibodies and recombinant proteins, they were able to show that M1 macrophages showed a greater migration towards the CM conditioned media. Again, with the use of neutralizing antibodies and recombinant proteins, they were able to show that cardiomyocyte-derived BMP proteins are involved in macrophage recruitment, at least in vitro. They were also able to show that cardiomyocyte-derived BMP4 is involved in the recruitment of M1 macrophages rather than BMP2. They also analyzed the receptor concentrations and found that both receptor subtypes were upregulated in M1 macrophages. Similarly, in modeling the impact of recruited M1 and M2 macrophages, they were able to show that exposure to M1 conditioned media resulted in an increase in cardiomyocytes, but a lower proliferative profile. Other studies have confirmed that PDGFR alpha is influential in the differentiation process, and this study confirmed that, showing that when cardiomyocytes were exposed to M1 conditioned media, they were PD. So really, there is been a bit of ambiguity in the literature on the role of the inflammatory environment on the injured myocardium. Initially, they thought that inflammatory environment was associated with the detrimental phenotype. However, other studies have shown that pro-inflammatory cytokines can have both beneficial and detrimental effects, really dependent on the overall duration and the concentration of the inflammatory process. This study did suggest that some of the possible mechanisms involved in increasing yield of cardiomyocytes are exerted by the pro-inflammatory macrophages. This was shown because of M1 macrophages showing when these induced stem cells were exposed to macrophage conditioned media, the canonical BMP signaling pathway was activated, as demonstrated by the phosphorylation of SMAD1/5. The study did conclude that macrophage-derived BMP proteins are sufficient to affect the biochemical signaling of cardiac repair cells, as was shown through the neutralizing antibody experiments and also the recovery with recombinant BMP. Being that M1 macrophages had the highest expression of BMP molecules, they are believed to be, at least in part, responsible for the overall increase in cardiomyocytes by affecting the differentiation of surrounding progenitor cells. So the beneficial roles of BMP proteins on cardiomyocyte differentiation is that you can look at the concentration of BMP4 to determine the stage of maturation of cardiomyocytes. Also, M1 macrophage-derived BMP proteins were able to rescue cardiomyocytes, corroborating the notion that the dose is crucial for the beneficial effect observed with repair cells. Other studies have used recombinant BMP2 and have recently shown to reduce. Another study showed that cells incubated with BMP4 before injury with hydrogen peroxide showed reduced cell death. And this study asserts that the positive role of M1-derived BMP proteins onto human mesenchymal cardiomyocytes could be ascribed to a protective effect against apoptosis, but also to induce differentiation of progenitor cells into cardiomyocytes. Lastly, I just want to go over some limitations that encountered in this paper. So as mentioned earlier, the expression of the BMP receptors was not measured before migration, and the authors suggest that that it was possibly modulated during migration or differentiation. So this is something that they would like to test in the future. Another limitation is really just the heterogeneity of stem cell differentiation. For the cardiomyocyte population that they were able to induce, they only were able to get a purity of about 76%. Therefore, they really weren't able to rule out contributions of other cell types in the contribution of BMP protein released into the conditioned medium. Also, as mentioned before, PDGFR alpha is a known precursor for smooth muscle cells and endothelial cells. So these could actually contribute to the vascular compartments of the chamber rather than the functional contractile units. For me, the major limitation of this paper was that they really did a poor job in describing their microscopy. They didn't have anything about the analysis they used, the software, or the setup. Another limitation that I found was that all data that they used, I feel like they could have gotten a lot more information if they'd used proper microscopy analysis software. I was also disappointed that they didn't try and quantitate any of the extracellular matrix proteins that are deposited. So I think that that would be a good follow-up study. That was my presentation. Thank you, everyone, and I wish you the best through this troubling time.